These are the structures and the description and the chemistry about the branched chain amino acids. These branched chain amino acids, BCAAs, they include three types of the amino acids. The skeletal structure, the backbone structure of these three are common. The alpha carbon surrounded by a carboxyl carbon, an amino group, and a hydrogen. The R chain of these branched chain amino acids contain aliphatic chain but in a branched manner. That's why they are called as branched chain amino acids. We have three types of these amino acids: valine, leucine, and isoleucine. Their R chain contains aliphatic chain but not in a linear way. There is a branch present somewhere, so they are branched chain amino acids. Valine, leucine, and isoleucine. These three are the examples of branched chain amino acids. So these branched chain amino acids, valine, leucine, and isoleucine, they are essential and they are required for muscles. Branched chain amino acids account for 35% of the essential amino acids found in muscle proteins and account for 40% of the total amino acids required by the body. As these branched chain amino acids are the essential amino acids and they are not synthesized in our body, so we must have to take these amino acids in our diet. So the total essential amino acids that are required by our muscles, out of these total essential amino acids, the 35% of essential amino acids should be branched chain amino acids. For muscle proteins and for our overall body functions, we required 40% of the essential amino acids in the form of branched chain amino acids. So these are valine, leucine, and isoleucine. These amino acids are required in greater amount as compared to other essential amino acids. The branched chain amino acids are the essential amino acids. They are required for muscles because they are the important component of the muscle proteins and the overall requirement for our body. The total essential amino acids should contain 40% of branched chain amino acids. These branched chain amino acids, they suppress protein breakdown and they prevent muscle wasting because they are the important component of the proteins present in the muscles, like myosin, like actin, like titin, and some other protein fibrils. So these branched chain amino acids, they suppress protein breakdown and muscle wasting. The wastage of the muscles is prevented in the presence of enough amount of sufficient amount of these branched chain amino acids. They decrease muscle soreness. Muscle soreness is the tiredness, the fatigue, or the inflammation of the muscles appear because of the prolonged or because of the chronic physical activity or a hectic activity or extreme exercise that leads to muscle cramps, muscle pain, and muscle inflammation. And it may last for 24 to 48 hours, usually when the body is deficient in branched chain amino acids. After facing a long-term exercise, after doing a severe or after doing a critical physical activity, prolonged physical activity, the feeling of tiredness, the feeling of fatigue, and the muscle cramps or inflammation in the muscles, the pain in the muscles, they are called as muscle soreness that can last up to 48 hours after the exercise. So if we are sufficient in branched chain amino acids, so the symptoms of muscle soreness are decreased. These branched chain amino acids reduce exercise-induced fatigue. They decrease protein breakdown. Basically, these branched chain amino acids, they enhance the synthesis of muscle proteins and they decrease the breakdown of these proteins. Normally, in a normal condition, our muscle proteins are in a balanced way. This is the protein turnover, the protein breakdown, and the protein synthesis are in a balanced way. In the presence of sufficient amount of branched chain amino acids, the protein synthesis in muscles, the synthesis of muscle protein is 30% increased. I'm repeating, in the presence of branched chain amino acids, the protein synthesis or the rate of protein synthesis in muscles is increased by 30%. So in the sufficient amount of branched chain amino acids, the protein turnover rate shift towards the synthetic or the anabolic phase. The deficiency of branched chain amino acid leads to muscle protein leads to the catabolic state, and in the presence of sufficient amount of branched chain amino acids, the protein turnover in the muscles shift towards the anabolic phase. So if we are taking the sufficient amount of branched chain amino acids, then we are, or our muscle proteins are in an anabolic phase. So these branched chain amino acids decrease protein breakdown, and because of the decreased protein breakdown in muscles during exercise condition, this leads to a decreased level of an enzyme which is creatine kinase, CK, which is an indicator for muscle damage. After heavy exercise, the protein breakdown is increased and your muscles may move towards the damage or the wasting. The muscle damage or muscle wasting is usually characterized by the increased level of enzyme creatine kinase. This creatine kinase is active in heavily exercising muscles. These branched chain amino acids, usually they are used as an efficient energy source during exercise. Most of the energy drinks and most of the protein powders, the protein drinks, they contains large amount of branched chain amino acids, and usually the person who are physically active, the sportsmen and some athletes, they are used enough amount of branched chain amino acids to prevent themselves from muscle wasting, from tiredness, from fatigue, and from muscle soreness. So these branched chain amino acids are the essential component of different protein rich diets. These branched chain amino acids are the essential component of whey protein. Whey protein, basically, this is a protein supplement which is obtained from the liquid portion of the milk during the manufacturing of cheese from curd. During manufacturing of cheese, the water portion or the liquid portion which is separated from the curd, it is called as whey. It contains different amino acids. There are different protein powders are available as a nutritional supplement. They are rich in branched chain amino acids. So these whey protein provides essential amino acids and they stimulate the release of anabolic hormones. Stimulates the release of anabolic hormones. So that this how they reduce stress. The exercise is induced stress on our muscles. They have hypotensive effects and they increased bone mineral density. So this whey protein is usually used by different sportsmen, different athletes, and those who are in heavily exercise conditions to prevent themselves from exercise-induced fatigue, exercise-induced tiredness, and muscle soreness. So these essential amino acids which are present in whey protein, they inhibit protein breakdown in the muscles, they stimulate protein synthesis in the muscles, they stimulate the release of some anabolic hormones like growth hormones like insulin. They reduces the stress which is induced by the exercise. They have hypotensive effect and they increase bone mineral density by enhancing the absorption of calcium and phosphate into the bones. The dietary source of branched chain amino acids: milk, egg, fish, beef, chicken, corn, chickpeas, lentils, whole meat, brown rice. The daily requirement should be 5 to 12 grams per day. And this requirement is increased in athletes. Up to 20 grams of athletes required branched chain amino acids: valine, leucine, and isoleucine. Best source of branched chain amino acids is casein, which is a milk protein. And from casein, we obtained whey protein also. For example, 1 gram of casein contains approximately 300 milligrams of branched chain amino acids. And cow milk, 100 ml, 1 deciliter of cow milk contains approximately 3 grams of casein. It means 1 gram of casein contains 300 milligrams of branched chain amino acids, and 100 ml of cow milk contains 3 grams of casein. So 100 ml of cow milk contains approximately 900 milligrams of branched chain amino acids. So if we are taking a cup of 250 ml, a cup of milk daily, it means this milk provides approximately more than 2 grams of branched chain amino acids daily. Hundred grams of beef contains 6.8 grams of branched chain amino acids. 1 gram of chicken contains 5.5 to 5 grams, and a single egg contains 1.5 grams of branched chain amino acids. So if we take a cup of milk, an egg, and a piece of a meat daily, then we could be sufficient in branched chain amino acids: valine, leucine, and isoleucine. Now the metabolism of branched chain amino acids. This is all about the functions, the dietary source, the importance of branched chain amino acids. Okay, now the metabolism of branched chain amino acids. Initial reactions of the metabolism are same. They share the common pathway. Initially, the common pathway are the 3 initial steps are the common in branched chain amino acids metabolism: valine, leucine, and isoleucine. The first reaction of their metabolism is transamination, means the transfer of amino group from an amino acid towards a keto acid that converts this amino acid into its respective keto acid, and that converts a keto acid into its respective amino acids. And for most of the transamination reaction, amino acid could be different, but the alpha keto acid is alpha ketoglutarate that converts into glutamate after receiving the amino group from the amino acid. So these transaminase enzymes are pyridoxal phosphate linked. The active form, pyridoxal phosphate, is the active form of B6. The second step is the oxidative decarboxylation, means the oxidation followed by decarboxylation. After transamination, these amino acids, leucine, valine, and isoleucine, converts into their respective alpha keto acids. Then these alpha keto acids, upon oxidation and decarboxylation, means the removal of carbon dioxide, removal of a carbon. Upon oxidation and decarboxylation, in the presence of dehydrogenases, these dehydrogenase enzymes requires their prostatic groups NAD, coenzyme A, TPP, thymine pyrophosphate, lipoic acid, NAD-CoA, TPP, and FAD. These all are the vitamin B complex derivatives, and the lipoic acid is somehow similar in structure and functions like B complexes, but this is not the actual B complex because it can synthesize in our body. This lipoic acid is a derivative of fatty acid. Lipoic acid is a derivative of fatty acids. It can synthesize in our body, so it is not a vitamin, but it has vitamin-like functions. So property-wise, it is not a vitamin, but according to the function, it is a vitamin because it is required for the activity of different enzymes. This lipoic acid is the prostatic group for different enzymes. Dehydrogenation is the third step in the metabolism of these branched chain amino acids. Dehydrogenation is the removal of hydrogen. It is catalyzed by the enzyme dehydrogenase or oxidase. The difference between the dehydrogenase of step number 2 and step number 3 is the step number 2 dehydrogenase required NAD-CoA, TPP, lipoic acid, and FAD as cofactor. But the step 3 dehydrogenase enzymes required FAD as cofactor. The step 2 dehydrogenase involves in oxidation as well as in decarboxylation, the removal of carbon dioxide. But the third step dehydrogenase enzymes are involved only in oxidation, not decarboxylation. So these are also called as oxidases, which are linked with FAD only, not with lipoic acid or any other cofactor. Okay, so these three are the common reactions: transamination, the oxidative decarboxylation, and dehydrogenation. Okay, now the first reaction is the transamination. Leucine, when combines with alpha ketoglutarate in the presence of enzyme transaminase, branch chain transaminase or leucine transaminase, this leucine becomes its respective keto acid, which is alpha keto isocaproic acid, and alpha ketoglutarate converts into glutamate. So the first reaction is the transamination. Leucine converts into its alpha keto acid, which is isocaproic acid. Valine, upon transamination, converts into alpha keto isovaleric acid, the acidic form of valine. Isoleucine, upon transamination, converts into alpha keto beta methyl valeric acid. These alpha keto acids are coming from the transamination of these branched chain amino acids. So the first step of the catabolism of these amino acids is the transamination. The transfer of their amino acids to alpha ketoglutarate that converts these amino acids into their respective alpha keto acids and alpha ketoglutarate into its respective glutamate. Okay, now after transamination, the second step is the decarboxylation. The oxidative decarboxylation. Basically, there are different reactions occurring. That's why these enzymes requires NAD, FAD, TPP, CoA, and lipoic acid for their activity. There is a mechanism of oxidation. There is a mechanism of decarboxylation. And there is the mechanism of addition of coenzyme A to these alpha keto acids which are coming from the transamination of branch chain amino acids. So after transamination, these alpha keto acids undergoes oxidative decarboxylation. The oxidation reaction, which is coupled by decarboxylation reaction, catalyzed by the enzyme dehydrogenases. The alpha keto isocaproic acid, which is coming from leucine, upon oxidation and decarboxylation, means the removal of one carbon and addition of coenzyme A, it becomes isovalerial CoA, one carbon less. Alpha keto isocaproic acid becomes isovalerial CoA upon oxidative decarboxylation in the presence of enzyme dehydrogenases or decarboxylases. Sometimes they are also called as decarboxylase. The alpha keto isovaleric acid, which is coming from valine, it becomes isobutyryl CoA upon oxidative decarboxylation. The oxidation, which is coupled by decarboxylation. The alpha keto beta methyl valeric acid, which is coming from isoleucine, it becomes alpha methyl butyryl CoA. These dehydrogenases or decarboxylases are clinically significant. Their deficiency may lead to different abnormalities, which we'll discuss in the later session of this lecture. The third step after the dehydrogenation and decarboxylation, the oxidative decarboxylation that converts the alpha keto acids into their respective decarboxylated coenzyme A form. Then they undergo oxidation or dehydrogenation, the removal of hydrogen group in the presence of FAD. They make these unsaturated derivatives of CoA. This is the removal of hydrogen, and these hydrogen which are removed from these CoA derivatives of branch chain amino acids, this hydrogen is accepted by FAD, and this FAD becomes FADH2. So the catabolism of branch chain amino acids gave FADH2. And in the upper step, in the previous step, step number 2, which is oxidative decarboxylation, upon the removal of hydrogen or upon oxidation, they produce NADH. In this step number 2, the hydrogen which are released by the alpha keto acids because of the oxidation is accepted by NAD+. And in step number 3, the hydrogen which is released by the aliphatic chain of these acids is accepted by FAD, that converts FAD into FADH2. So these branch chain amino acids, first they catabolize through transamination, then they undergoes oxidation and decarboxylation. So their oxidation gives NADH and FADH2 a little amount of energy. So leucine upon transamination, oxidative decarboxylation and dehydrogenation, it becomes methyl crotonyl CoA. Valine upon transamination, oxidative decarboxylation and dehydrogenation, it becomes methacrylyl CoA. Isoleucine upon transamination, oxidative decarboxylation and dehydrogenation, it becomes triglyl CoA. These are the common 3 reactions for the catabolism of branch chain amino acids. But if we see the end product after these 3 reactions, they are not the end product that we discussed in our first lecture of the reactions of amino acids, in which I said the amino acids converts into 7 intermediates or the 7 end products. Either 1 or 2 could be the end product of these amino acids like acetyl-CoA, like pyruvate, propionyl-CoA, succinyl-CoA, fumarate, acetoacetate. So these are not the end products, so it means they further catabolized. But the initial three steps are the same during the catabolism of these branch chain amino acids. They converts into their respective alpha keto acid and their CoA derivatives. Upon further metabolism, they give their final end products. So leucine, valine, and isoleucine, we have three types of branch chain amino acids. Upon transamination, they become their respective alpha keto acid. Upon oxidative decarboxylation, in the presence of enzyme dehydrogenase or decarboxylase, they becomes their CoA derivatives. Leucine gives isovalyryl CoA, valine gives isobutyryl CoA, and isoleucine gives methylbutyryl CoA. After oxidation or after dehydrogenation in the presence of FAD-linked enzyme, we obtained methylcrotonyl CoA from leucine. Then upon further catabolism, some hydrolysis and cleavage of different parts of this aliphatic chain of these alpha keto acids in the presence of biotin, again a vitamin B complex B7 biotin. In the presence of biotin, the further reactions are proceeding that finally leads to the formation of acetoacetate plus acetyl-CoA upon the catabolism of leucine. So the final end product of leucine catabolism is acetoacetate and acetyl-CoA. The valine, after transamination, oxidative decarboxylation, and dehydrogenation, it further catabolized into propionyl-CoA. Again, in the presence of biotin, B7, it gives succinyl-CoA. The end product of valine catabolism is succinyl-CoA. Isoleucine, again it gives two end products. It gives acetyl-CoA, then the remaining chain can form propionyl-CoA. This propionyl-CoA ultimately gives succinyl-CoA. So the first three reactions are the important and common in the catabolism of these branch chain amino acids, leucine, valine, and isoleucine. And upon further catabolism, in the presence of biotin, leucine gives acetoacetate and acetyl-CoA, valine gives succinyl-CoA, and isoleucine gives succinyl-CoA and acetyl-CoA. So here we can differentiate these amino acids as glucogenic and ketogenic amino acids. If they give the intermediates of glucose metabolism, then they are glucogenic amino acids. And if they give the metabolic intermediate for ketone bodies that are associated with the formation of ketone bodies, then it is ketogenic amino acid. So leucine gives acetoacetate and acetyl-CoA. Leucine is a ketogenic amino acid. Valine gives succinyl-CoA that can convert into oxaloacetate during TCA cycle, and this oxaloacetate can be utilized for glucose synthesis. So this valine is a glucogenic amino acid. Isoleucine gives succinyl-CoA as well as acetyl-CoA. So isoleucine is glucogenic as well as ketogenic amino acid. Acetoacetate and acetyl-CoA, these two are the structures that contain ketone body, that contain ketone group in their structure. So they are called as ketone bodies. So the amino acid that gives a ketone body, it is called as ketogenic amino acid. Or if the amino acid upon catabolism give intermediate of glucose synthesis for gluconeogenesis, then it is a glucogenic amino acid. Catabolism of branch chain amino acids produces more glutamine and alanine in muscles to be transported into the liver for urea cycle through glutamine pathway or through alanine cycle that we have discussed already in our previous lectures. The transport of nitrogen from muscles and from tissues towards the liver for the urea cycle, it occurs through two pathways, through glutamine or through alanine. So the catabolism of branch chain amino acids produces more glutamine and alanine. Metabolic disorders that are associated with branch chain amino acids. The first and the most common or the important one is the maple syrup urine disease, MSUD, or it is also called as branch chain ketonuria. The accumulation of branch chain ketone acids in blood as well as in urine. Branch chain ketonuria, the keto acids that are coming from the catabolism of branch chain amino acids that are accumulated in the blood as well as into the urine. Because of the deficiency of the enzyme decarboxylase or dehydrogenase that catalyze the step number 2 of their catabolism. After the first step, which is the transamination, these branch chain amino acids convert into alpha keto acid, alpha keto isocaproic acid, alpha keto isovaleril acid, methyl valeryl acid. These three alpha keto acids are coming from the transamination of leucine, valine, and isoleucine respectively. Then these alpha keto acids undergo next step, which is the oxidative decarboxylation, oxidation, decarboxylation, and addition of coenzyme A. This step number 2, which is catalyzed by the enzyme dehydrogenase that requires NAD, FAD, lipoic acid, TPP, CoA for their activity. So the deficiency, the abnormal synthesis of this enzyme leads to defective metabolism of these amino acids. There is increased concentration of alpha keto acids into the blood because these alpha keto acids are not further metabolized into their CoA derivatives due to the lack of the enzyme decarboxylase. It is an autosomal recessive disorder because of the mutation in the gene that synthesizes decarboxylase enzyme or dehydrogenase enzyme. So the accumulation of alpha keto acids that are coming from the transamination of these amino acids. Okay, now the symptoms, because of the accumulation of alpha keto acids that are coming from the transamination of branch chain amino acids, leads to the accumulation of again these branch chain amino acids. Because the transamination reactions are the reversible pathways, so these alpha keto acids can be converted back to their respective amino acids. When the equilibrium between reactant and the product of the transamination shift towards the product, the more product is formed because the product is not further converts into their respective intermediates. So this product can be converted back into its respective substrate. These transamination are the reversible reactions. So in this condition, when decarboxylase enzyme is deficient, there is the accumulation of alpha keto acids of branch chain amino acids. This accumulation leads to again the conversion of these alpha keto acids into the branch chain amino acids. So there is the accumulation and the disturbance of amino acid pool in the body. The amino acid pool and the overall protein turnover rate is changed. And also these alpha keto acids starts to accumulate into the blood, as well as they are excreted out into the urine. So the symptoms include the eating disorders. Because of the eating disorders, abnormal feeding behavior, the symptoms of vomiting appears. This vomiting leads to dehydration, as these alpha keto acids that are coming from the transamination of these amino acids, they are acidic in structure, so their accumulation leads to metabolic acidosis, a decline in the pH, because these are acidic in structure. As these alpha keto acids are elevated in the blood, so they excreted out in the urine. The urine, which is concentrated with the alpha keto acids of these branched chain amino acids, have a specific odor, a characteristic odor, which is like a maple syrup. Maple syrup is a syrup coming from the heating and burning of the sugar. When the sugar or the saturated sugar is burned, there is a specific or the characteristic odor. The smell of this syrup is called as maple syrup. The burnt sugar syrup like smell of urine of these patients. So this is the unique property of the patient that are suffering from the maple syrup urine disease. The characteristic of this disorder is the urine of these patients has specific odor like maple syrup because of the increased accumulation of these alpha keto acids. If this remains untreated, it may lead to mental retardation and physical disabilities, because they are keto acids, they lead to metabolic acidosis. So there is increased chance of mental retardation and disabilities of physical activity. The next is the continuation of maple syrup urine disorder. The previous one was the genetic mutation or the genetic fault of decarboxylase enzyme. The next one is the intermittent branched chain ketonuria. Again, this is the accumulation of the alpha keto acids because of the defective enzyme decarboxylase. But in this condition, the synthesis of decarboxylase enzyme is normal without any mutation, without any defect at gene level. But the activity of this decarboxylase is decreased because of the deficiency of its prosthetic group. The lipoic acid, which is derived from the fatty acids, and it is required for the activity of this enzyme. So the abnormal metabolism of lipoic acid or the deficiency of lipoic acid leads to the decreased activity of decarboxylase. Again, there is the accumulation of these alpha keto acids into the blood as well as into the urine. The symptoms are the same, but it is not congenital, it is intermittent. It may appear at any stage of the life because of the nutritional deficiency. So because of this, there is an impaired oxidative decarboxylation reaction. This lipoic acid is required for the activity of dehydrogenase enzyme that catalyze oxidative decarboxylation, like in the branched chain amino acids. And when you will learn the glucose metabolism in your GI module, the glycolysis, the TCA cycle, then again you will learn about the oxidative decarboxylation of different intermediates during glycolysis and TCA cycle. They also require lipoic acid for their activity. So the deficiency of lipoic acid leads to decreased activity of oxidative decarboxylase enzyme, which are required for the oxidative decarboxylation of alpha keto acids, of branched chain amino acids, pyruvate, and alpha ketoglutarate. The pyruvate and alpha ketoglutarate are involved in the glucose metabolism, and their symptoms appear later in life because it is not a genetic disorder, it is a nutritional disorder, and it can be treated by giving lipoic acid supplement and some other supplements because it is intermittent. It appears gradually. The last one is the isovaleric acidemia. Because of the deficiency of the enzyme isovaleryl-CoA dehydrogenase, the third enzyme of this pathway, the leucine degradation pathway that converts isovaleryl-CoA into its respective oxidized form, unsaturated form. So the defect of this enzyme leads to the accumulation of isovaleryl-CoA. This isovaleryl-CoA is hydrolyzed into isovaleric acid instead of acetoacetate or acetyl-CoA for leucine degradation. Again, there is the accumulation of isovaleric acid. So it is characterized by acidosis and vomiting. If untreated, it leads to coma. So you can read this topic from